All right, welcome back to the science lab here. So we're just kind of getting set up, but what I'm going to have us do today is we're going to take a look at cheek cells, our own cheek cells, actually. So I did this with my students recently. We're doing a cell biology unit, and it's easy to look at, prepared slides of cells from other organisms, but it's kind of neat to see your own cells. So this is a fairly simple thing to do. But you would need a microscope to be able to do this. You would also need some kind of a slide. We're going to be using traditional glass slides, but you could use like a plastic slide hypothetically, but getting the microscope is really going to be the major key.
And most of the things I've noticed on the show when we do science experiments, they can make at home. So they could probably make a microscope at home then.
Yeah, so what I was going to say, we really do try to do these things that you guys could also replicate, but you can't purchase a lab-grade microscope. There actually are some cool ways to approximate a microscope, including using pieces of a laser pointer. So if you're interested in making a cheap microscope, look into either DIY microscopes, just like type in DIY microscopes to YouTube or Google, you get lots of suggestions. Also, you could check out this thing called a fold scope, which I may talk about on the show in a few weeks, possibly. All right. Okay, so what we're going to do, guys, is we're going to prepare microscope slides, and I'm briefly going to explain how microscopes work. Have you guys check out your own cheek cells, and we're going to try to actually photograph them through the microscope lens. Okay, cool. Using your smartphones. And this is something that I've actually seen videos of lab scientists doing, just to get a quick shot of whenever they're looking at. Okay, so the first thing I need you guys to do, if you could each take a glass slide for me. You can just drop that, Nate. I'll put it in when we got a second.
See who brushed their teeth this morning.
Okay, we're not gonna get that gross. I promise. This is it's simple. So go ahead and just put them down on the
All right.
desk there. So what I have here is methylene blue. This is a dye that we use to dye different specimens. If we were to look at our cheek cells plain, we wouldn't really be able to see them very well because they're largely translucent.
Right.
So you can pull up a lot of this blue stuff here, but you really don't need much. So you just really need to touch, you don't even need to touch this thing, just touch the end of the dropper to the center of your slide.
Is this just, you said this is a dye, so it's not like just food color?
Or a stain, I should say.
Okay.
Yeah. It's a stain a specimen stain for biological slides. That is all you need, Nate.
Wow. Maybe dip it back in there. Oh, that's probably good.
Like that?
Yeah, those are really nice little tiny drops, guys.
Thank you.
That's perfect. You guys are so talented.
Oh.
Okay, so if you could each take a flat toothpick from my box here. Okay, and then gently, what I need you to do is just scrape the inside of your cheek with the toothpick.
All right.
With the tip of the toothpick, specifically.
Oh.
I don't know if I got it.
Oh, either side. Yeah, no, it doesn't feel like much, but if you scraped it with the end, you did. So then all you need to do is dab that end in your little blue dot of methylene blue. And I'm just going to plug these microscopes in.
I don't feel like I did it right. Hang on.
Do you want to try again? You're welcome to grab a new toothpick. Yep, and you just kind of dab it in there a couple times. You dab it in there, Nate?
Yeah, I did.
If we have any kind of an issue, we can always try again.
Okay.
You guys really did do small. I'm always like really on my students about tiny dots, but to them, like a tiny dot is just like so much of it.
Right.
You guys took me really seriously. Okay, so what I have here are glass cover slips. These are really thin sections of glass.
Place this.
And what we're going to do, microscopes really see in an almost two-dimensional plane. They don't see 3D very well, and so you'll notice as we look at these things, we'll be seeing sort of things in a 2D plane. If there's cells that are kind of rolled on themselves, parts of them will appear out of focus. Okay. So this will help us a little bit to flatten them into a 2D plane. So go ahead and take that.
Oh, is that because of like the precision of the lenses?
Yeah, exactly.
Really...
Exactly, just because of the focus.
Right.
And you can see in 3 dimensions, but only a 2-dimensional plane is really
kind of like
focus.
when we take a picture, right? And like you're in focus and everything else is a little bit blurry.
Exactly. Exactly. Okay, so just cover your blue dot, and you should see it spread out a little bit as you do that.
Yeah.
You can push it down just to get it to spread a little bit more. Okay, cool. So, and And we can just, I'll just have you guys wait to look at these for just a second. I was just going to explain really quickly how microscopes work. So microscopes have two kinds of lenses. They have the optical lens, that's the part that your eye is going to go to, and they have the objective lenses down here, which increase the power of the optical lens. And it's really simple to figure out the power of an objective lens. You can just look on it and it tells you the magnification. So like this one, the first number here is 10. So there's a 10 times magnification from that guy.
Okay.
This guy here, you see it says 4, so it's a 4 times. And this guy here,
40.
40.
It's a big one.
The optical lens provides 10 times magnification. So the combination of the two lenses actually multiplies. So if we have the optical lens and add the smallest objective lens, which has a power of 4 times, we would have a total magnification of?
What was the first one?
10 was the optical lens.
40.
So 40,
Okay.
right? So 40, and then as we bump up to 10, we get 100, and as we bump up to 40, we get 400
Wow, okay.
times total magnification.
Yeah.
Okay, so let me spread these out for you guys, and I'm here to help you as your biology teacher, but I'm going to try to walk you through this. Okay, so the other part of the microscope that we should just think about is the stage. So the stage can be raised and lowered by these focusing knobs on the left here. The largest focusing knob is called the coarse focus. The coarse focus focuses it makes big changes to focus. Down here, this smaller knob is the fine focus, right? It makes smaller changes to the position of the stage. So we'll use that second. So what I want you both to do is, could you set first set your objective lens to the lowest power.
Lowest power?
Yeah, and we always want to start on microscopes at the lowest power objective lens because that gives us the broadest view, and that makes it such that we can sort of see what we're looking at, and then we can zoom in from there.
Right, because if you zoomed in a lot and you were just like had a lot of blue, you'd be like, I don't know which direction to go.
Right, and we can often see clumps of cells as we look from above, and then we can pick, that looks like a good clump, we can zoom in. Okay, so now you can use the little clips to secure your microscope slide on the stage. And you can use those little clips just to cover it up. They're sort of handy for just keeping the slide in place. Okay, and then what you want to do is you want to look through your objective lens and move the coarse focus back and forth until you see what you should see are a lot of blue dots, which are your cells. At the lowest focus, the cells really just appear like the blue stained dots. But as we get closer, we should be able to see some difference. So if you get close to focus, use the fine focus to bring it even closer into focus.
Okay.
And then Nate, when you get a set, can I also just see what you're looking at? So what we're going to try to do, Homework Hotline viewers, to let you see what we're seeing, is we're going to actually take a photograph of what we're seeing in the slide. We'll send it to our own Gmail, and then we'll have Darren, who's standing patiently at the social media board, pull that picture up for us.
So there's a needle on it,
Is it
and
just
is that
a blue dot?
is that for size, or is that like to give you reference for size, or is that to
So no, that's for
line up
location
the
position.
center? Okay.
So that's the line at the center. So what you really want to do is push the part of the specimen you want to see most to the end of that needle. That's the center right there. So as you zoom in from there, whatever you positioned right in front of that will be at the center.
That way I don't lose it whenever I'm zooming.
Exactly. Can I just take a look and see where we're at with yours?
So we have some like dark blue dots.
Oh yeah,
and those are my
the dark
cells.
blue dots are your cells. That is perfect.
All right.
Okay. Fantastic. And we're not going to take a picture at the lowest zoom because it's just not that interesting yet.
Yeah, it literally just looks like a blue kind of like spill with some darker blue dots on it right now.
Exactly
just
right.
So you know.
Becca, have you gotten your little blue dots?
I think so.
Can I check just to see?
Yeah.
Let me just double check. Science teacher.
Okay.
All right. Oh, we got here. Oh yeah. Oh look, did you already zoom in?
Yeah, I zoomed in.
Okay, Becca, overachiever. Okay.
She's like that student who's kind of ruining your lesson because she already
did
figured something out.
everything. I'm
That's
just like,
that's yeah.
yeah, Becca, you did not
Sorry.
get many cheek cells, but that is definitely one right there. Okay, so can we try to get that one? Okay, fine, Becca, you win. All right, so Becca's got one of her cheek cells pulled up into pretty good focus at the highest magnification. So Becca, what I'll have you do is just adjust the fine focus a little bit, try to get that perfect position, and then I'm going to talk to you about photographing. In the meantime, Nate, thank you, patient students.
I follow instructions.
Okay, that's right.
Good job.
So Nate, what I'll have you do now is, can we bump up to the 100 times
All right.
power?
So we're just going to turn this a
Yep,
little bit.
just rotate
We're looking
it.
for the
You're rotating it clockwise, I
This
guess.
way,
I
right?
lost it.
Because this is the
There we go.
400 power.
That's exactly right. So, and we just want to bump up one power at a time, again, just to make sure, okay, we see what we're seeing, move it to the center, move the part of the specimen we want to see to the center.
So like now it's a little off the needle, so I kind of want to move it a bit
Yes,
more.
exactly.
Right.
So now just adjust gently your slide. Becca, thank you for waiting patiently over there. We'll be with you in just a moment.
No problem.
All right, there we go.
Okay,
Center
fantastic. Let me just take a look here. Beautiful. Okay. And I'm even thinking that maybe, yeah, these slightly more clear ones. Oh,
Yeah.
that's a perfect.
Oh yeah.
Okay, so one of the things,
cool.
Becca, your cell is a little bit folded. So one of the things, Nate's got one here, you can see the nucleus perfectly.
See, because I
That's
waited
cool.
and followed instructions.
So Nate, take a look there at that.
right.
And again, we're just at 100 times power, so we're going to bump it up to 400 times power, take a couple pictures, send them, see them, and then, of course, get back to the normal thing. So Nate, bump up to 400 times.
400. So one more clockwise.
One more clockwise turn. We're going to bring that into focus. Can you still see that guy there? Can you see the
yeah.
nucleus?
Uh-huh.
Yeah, really, this is a good cell. Do you need to use the fine focus at all to bring it
think
slightly
so.
more into focus?
Yeah. Eyelashes are getting in the way.
Yeah, and it's sort of tricky getting the feel for using a microscope. It's a little awkward at first, but it really is something that if you practice it a little bit, it starts to feel more natural. I have taken to just looking at lots of things under the microscope, and sometimes it's a total failure, like the microscope doesn't reveal much. Other times it's cool, and I'm getting better at using it. Okay, so now photographing. This is the
Alright.
tricky thing. So smartphones are actually surprisingly good at capturing these images. I have an image right here, which I'll just show to you guys. So there's a level of focus that we can get. So
Okay.
pull up your cameras, and what we're going to do is we're going to actually just point the camera right down the barrel. It cannot be directly against the piece here. It needs to be a little ways away, and you're kind of, yeah, Becca, do you see how you can kind of get that circle to grow a little bit?
Oh yeah.
Did
yeah.
you see that?
Yeah.
So this is another thing that I have gotten good at because I have a bunch of students and I was helping all of them take pictures. So I practiced like 35 times. This is really tricky to do at first, but once you get a good shot, you can really see pretty well.
This
These
is
are
way
sort of-
harder
Nate,
than
that's-
taking a selfie.
So one thing we can also do, if you can just hold it steady, you can get a partner to come over and focus and to snap the picture.
Oh, there
Oh,
we
see
go.
that is, do you see that, Nate?
Could I do a video? Is that
Yeah,
possible?
why don't you try a video and see if you can
then I
get
Try
it.
a video because then you guys can see how difficult this is. But it's possible. I see it. I just have to get the right.
So both, oh, see, that's perfect right there. Okay, I know that we're making a lot of noise over here and you have no idea what's going on. Just a moment.
There we go. I got a good one.
Okay, you want to try to send that to Darren over there and we'll have him pull that up. Becca, you're so close. Could I help you?
Yeah.
Let me just see. So, oh, that's so perfect. Okay.
There we go.
It's so hard to.
It's really hard to hold steady, isn't it?
So yeah, if you do the video, you can do a screenshot.
All right. So Nate's got a good screenshot.
Oh my gosh.
See that level right there, Becca? That's what you want. But it's very hard to hold steady, and it's really, you have to be very precise.
Okay.
And it's a little bit frustrating, and several of my students were not happy with me when I tried to have them do this.
Yeah, the video makes it a lot easier, especially these high resolution
I might try
HD,
that
and then you
because
can
this
just
is
take a
just.
screenshot of it.
That's fantastic.
So it's homework at emilygriffith.edu.
Homework at emilygriffith.edu if you want to send in a question.
Okay. Okay. I'm gonna try a video.
I want to see how fast you can get this.
So I just,
Wait.
my phone just realized.
Okay.
But let me see, I can try it with your phone.
many.
I'd like to try.
Homework at
Too many cell pictures.
Yeah. It's going to make a selfie joke, and I guess I'm making it now, but
That is so good. All right, Darren, so hopefully that'll come
on
through
recording
in a
right now.
minute there. Oh, you got it on record.
Yeah.
All
Okay.
right.
One more time. I'm not very good at this.
oh.
Now, one of the tricks is this thing moves.
And using that as kind of a stabilizer.
That's what I was thinking. Is maybe. Oh, there we go. I think I can get it.
You snapping it? Oh, hold on.
Oh, Nate, push the button.
Now let's see. Oh yeah, I got a pretty good one on Nate's. I'm
There we go.
gonna text it along here.
I got one.
All right. All right. Can you send it? Can we
Like
send
we're
it?
fishing or something.
All right.
caught
So
it.
Almost.
I sent another one from Nate, assuming we got the.
Okay, so I'm sending it to.
And I emailed it. Should I have texted it?
I just texted it to that email address.
Okay.
Okay. So I'm going to email it to.
Interesting.
I'll text it also.
You double check the spelling.
Oh, I got it right there.
Yeah, that's
Oh,
gonna be it. That's
Homework
what I
Hotline
that's the one I
saves.
used.
All right.
Okay, well,
Okay, share.
Oh, you know what? I'm out of data. I should connect to Wi-Fi because this will take an hour.
the slight technical difficulties, folks.
I need to upgrade my.
We have some really great pictures here. I just
iPhone
wish you
plan.
could see them.
I don't know if mine's that great, but okay, where am I sending it to?
To homework at
I say this every day. Homework at Emily Griffith.
If only there were some sort of screen that had all of our contact information.
Right.
So,
we
you know what,
could-
and also what I will do, I have a really great shot of a cell that
Yeah,
I
mine
will
is
send
still
along
sending.
here.
Okay, sending. Is it there?
Is it 2 F's in Griffith?
Yes.
.edu. Okay, I also texted one of mine, so hopefully one of those will come through.
It's not coming through.
Yeah, so I can maybe. Well,
Yeah, mine is still
I
sending.
might try to
because of my
text it
terrible
in a
data.
second. So what was our? Try to text it to
The
that
address?
might work. So let's text it. 970. What was it again? I say it every day.
And we can call it in and talk about it.
And so-
970.
what we could also try to do is get this sorted out. We could
1666?
go to commercial,
Oh,
jump
here we
back.
go. So
there you
680,
go.
1666. Okay. Watch us at Homework Hotline struggle with technology.
It's tough sometimes, guys.
This is
It
how we
really-
know we're getting old.
It really is
Yeah.
tough.
Okay, I'm texting it now as well. There we go.
I think that might
Sent.
have sent.
It said sent.
Nope, it's sending.
So where do we get the text? They go to, okay. Texts just go to our email or our Gmail. So if you're texting us, it just goes straight to the Gmail. So we're refreshing right now.
Okay, well,
Maybe it's been
let-
reporting
let's-
as spam.
And let's maybe do this while we're trying to figure this out. Do you think is there a chance that we could just show what you guys have on your screens to a camera just briefly? It won't look
Oh,
fantastic.
yeah, we can do that.
But let's try to do that. Let's try to do the close-up camera, or maybe right here. If you guys want to go carry that up to
I don't know
Jeff.
if I'll
and
be able
let's-
to reach because I'm
Oh, you're
connected to
tied.
a lot of wires, but
Oh, that is so true.
Oh, there's,
should I?
get out of here.
It looks good. Oh yeah.
Get
so there
outta
you can
here.
See, you can see Nate's, but
just trying to hog
still
the
just
camera.
not quite in focus.
Do you see that little blue dot?
Okay.
See that little blue dot?
Well,
There it is.
You know, they'll probably come up as we're answering more questions.
yeah.
we
So
can-
hopefully it will. So I tell you what we're gonna do, guys. We're gonna jump back to the homework, or we're gonna go to commercial break. Then we'll jump back to the Homework Hotline studio. And when we do that, we will hopefully have a few cell pictures to show you. See you in a minute.
So we did actually, my awesome cheek cell picture came through. So let's show you it. Don't judge my photographer skills. It's not the best. But that right there, you guys, is my cheek cell.
And I'll actually just jump in. It's actually, I think it looks like more than one cheek cell sort of folded on itself. So what you often get is
Oh, so-
multiple cells kind of together. And so definitely the fact that it's darker. If we could jump to Nate's screen, beautiful picture, Becca. Not to take anything away there. But if we could jump to Nate's screen possibly. Nate also took a picture. His came through in his own Gmail. And you can actually see the nuclei of the cells in his.
Oh, his is way cooler. He has way more of them on there. I
Yes,
just
you
had
can
one.
see more of that, like the darker places are there's going to be more tissue folded over, but that one cell right in the center just below that centering needle, you can see sort of the outline of the actual cell. You can see little dots inside that are going to be different organelles, and you can see that large black center or darker center. That's the nucleus of the cell. That's the nucleus of
okay.
Nate's cheek cell. That contains all of
Very
Nate's
cool.
DNA.
So you're saying he didn't brush his teeth this morning.
Was that what it was? It's that
I just
Becca's-
have more DNA.
Becca's mouth is just super clean. Is that
It's so clean that just
No cells.
So, well, anyway, so a little bit of technical difficulty, but it really is kind of cool to see your own cheek cell, guys. So if you ever have a chance.
I didn't know that you could take a picture, a cell phone picture through that.
Yeah, and again, it is tricky, and there are some technological problems that can come up, but it's pretty good. And I've actually gotten some really great shots for class that I've used on different things that we've looked at under the microscope. So
definitely
it's
useful.
working for me, at least temporarily.
Yeah, you seem like you got that pretty dialed in.
Yeah, I'm a little bit better than I was when I started.
Yeah.
Practice makes perfect.